Hi, my name is Dr. Perran STOTT-ROSS, and I'm working as part of the Australian Grains Pest Innovation Program in Australia, working with Ari Hoffman and Paula Miner at the University of Melbourne. And so today I'm going to be talking about some of our work in aphids, where we're using endosymbiotic bacteria as a way of hopefully improving crop agriculture. So aphids, they can cause damage to crops in two main ways. So through virus transmission and direct feeding damage. And so turnip yellows virus is a really important aphid-borne virus in Australia, and it causes pretty substantial damage to canola. So it's transmitted by different aphid species when they feed on infected plants and then feed on a different plant. And so it causes yield and quality losses in canola crops and pulses. And so aphids can cause damage also by feeding directly on the plants. And so that takes away nutrients from the plant, it can stunt its growth. And so you can see on the right that there's a canola crop that's really heavily infested by cabbage aphids. So in Australia, aphids tend to only produce asexually, so that's without mating. And so aphids, the female aphids will give birth to live offspring without the need for mating. And so this makes them able to increase their population size very rapidly and quickly overwhelm the host. So when a plant becomes too crowded or unsuitable, the aphids can develop into a winged form that can then fly to other host plants and infest them as well. So the green peach aphid has a really wide host range, and that includes many different weeds. And so this allows them to persist outside of growing season. So in Australia, the green peach aphid is a really big problem because it can cause both direct feeding damage to plants, and it can also transmit viruses. And so in Australia, it's an important vector of the plant virus, turnip yellows virus, and this causes a lot of damage to canola. So in Australia, there are several different approaches to control aphids, and that includes biological control, where there's you can release different predators such as hoverflies into crops infested with aphids that will attack and eat the aphids. There's also parasitoid wasps, which can lay eggs inside the aphids, which will then develop into more wasps. And there's also things like fungal pathogens. In terms of other approaches, there's also chemical control, which of course is becoming less effective over time as green peach aphid now has resistance to a lot of different classes of insecticides. And they fall into two different categories. So you can have target site resistance and also metabolic resistance. So this talk will focus on a new type of aphid control, which targets the endosymbiotic bacteria that live inside aphids. And so there's two main categories of endosymbionts. There's obligate ones and facultative ones. So the obligate ones are basically found in all aphids. So they're essential to the aphid survival, and they provide essential nutrients to the aphid. And so there's the aphid can't live without the endosymbiont, and the endosymbiont can't live without the aphid. And these endosymbionts are transmitted from the mother to her offspring, and usually not between species. There's also a second type, which is called facultative, and these aren't required by aphids to survive. And there's a lot more diversity in terms of the effects that they have on the aphids. So they can have beneficial effects on the aphid or harmful effects on the aphid, depending on the environment. And so, like the obligate ones, they're transmitted from mother to offspring, but they can also be transmitted between different individuals or to different species through the environment. So for instance, if they share the same plant, they can be potentially transmitted between aphids that way. So the endosymbionts within aphids are really diverse in their effects, and they can either be beneficial or costly. So they can provide really amazing advantages like protecting aphids against parasitoids or fungal pathogens. They could even change the body color of the aphids. These effects really, they seem to depend on the environment that the aphids are in. So they might have a beneficial effect on a particular host plant, but under some other conditions, like if it's at a high temperature or there's insecticides, it might be a disadvantage. So there might be some situations where they're more of a burden. So endosymbionts are really interesting, but what can they actually do for us? What benefit do they have for agriculture? So there's a really good example for human health, at least, where Wolbachia are being used to control dengue transmission by mosquitoes. So if a mosquito, normally a mosquito is really good at transmitting dengue and malaria and other diseases, but if a mosquito carries Wolbachia, their ability to transmit dengue goes down a lot. And so if you release mosquitoes with Wolbachia into the wild, then you can almost eradicate dengue from some locations. So there's been a lot of field trials around the world now. It's had a huge impact on dengue transmission. In terms of agriculture, there's a lot of potential ways in which endosymbionts can be beneficial. So, for example, they can potentially reduce virus transmission by insect vectors. You can potentially change the sex ratio of beneficial insects to make them more efficiently produced. So if you've got some parasitoid wasps that are mostly male or equally male and female, you can change them so that they're now mostly female, which of course are the ones that attack the aphids. You can also potentially crash populations of pests by reducing their fertility or survival, or changing how they respond to temperature or insecticides. So there's a really good example of applications of endosymbionts in agriculture, and it comes from plant hoppers. So when you introduce an endosymbiont called Wolbachia into a particular species of plant hopper, they're no longer able to transmit a particular virus called rice ragged stump virus. And this, of course, causes a lot of damage to rice crops. And this particular bacteria can spread through the populations of plant hoppers. And then when the population has this bacteria, they can no longer really transmit this virus. And so there's a lot less damage to the rice plants. So to see how endosymbionts could be useful for crop protection in Australia, we started by doing a survey of these endosymbionts in aphids in Australia. And so we did a lot of field surveys. We collected aphids from crops and also just our backyards. And we looked for these different species of endosymbionts. And so this here shows 3 target species that we're interested in. And so one of these is the green peach aphid, which of course is a big problem for canola. And we found that it doesn't have any of these secondary endosymbionts. So almost all of the aphids just carry this obligate one called Bucknera. But in other species that aren't a big problem for crops, we found a lot of these other endosymbionts like Serratia and Hamiltonella. And so we can potentially use these to modify our crop aphids, pest aphid species. So there's 2 ways that you can do this. The first one is that you can use chemical treatments that target endosymbionts inside the aphids. And so this is basically an alternative to the insecticides that we're currently using, where you're targeting, instead of targeting the aphid, you're targeting the endosymbionts that live inside the aphids. And since aphids need these endosymbionts, you can eventually wipe them out over time. The other way to manipulate endosymbionts is to transfer them between species with microinjection. So you can take an aphid that carries an endosymbiont that you're interested in and transfer it to another aphid with a very, very small needle. And so this is what we've been doing in our lab, where we're taking endosymbionts from different aphid species, such as the rose aphid and the pea aphid. We're getting this really tiny needle and we're sucking out the aphid juice, and then we're injecting it into the green peach aphids. And so we've now introduced a few different endosymbionts into the green peach aphid, and they have some really dramatic effects. So for instance, this one called Rickettsia, it changes the body color of the aphid. So instead of being a pale green, they become a much darker green. And so this might not be that useful, but we're exploring all the different effects of these endosymbionts in terms of how they interact with parasitoids and predators. So this same endosymbiont also has a really, really big effect on reproduction of aphids. So if an aphid is, a normal aphid will produce a lot of nymphs in their lifetime, but if they carry this Rickettsia endosymbiont, the number of nymphs that they produce is cut in half. And so since they also have a shortened lifespan, this sort of shifts the timing of their reproduction, so they produce them a lot earlier on. We're also looking at how different aphids, when they carry these different endosymbionts, are able to transmit important viruses. And so this is an example that we've looked at in oat aphids. And so they're an important vector of a virus called barley yellow dwarf virus. And we basically looked at the ability of these aphids to transmit viruses when they carry this endosymbiont versus when they don't. And you can see by this figure on the right that the amount of virus that they transfer to plants is actually higher when they have this Rickettsia endosymbiont. So this isn't really that useful to us because we were hoping to find the opposite. So if we found an endosymbiont that reduced virus transmission, that could be really useful because you might be able to stop the spread of these viruses in the wild. So we're also looking at different chemical treatments. And so these really have the potential as an alternative to the current insecticides that we're using, because instead of killing the aphid, we're getting rid of the endosymbionts, and that sort of kills them, but it's over a longer time. So the first generation might not be effective, and the next generation is, because they can no longer reproduce. And so this is an example with a particular antibiotic where we've exposed aphids to these antibiotics. And you can see that this is the amount of the Buchnera endosymbiont inside the aphids. And it doesn't have any effect straight away, but in the next generation, the amount of Buchnera inside the aphids really, really drops off in that next generation. And so, of course, when you have none of this Buchnera, the aphids become infertile. And so this is a potential way to wipe out aphid populations across different generations. So we're also looking at other alternative chemical treatments like fungicides, and these have also got some really interesting effects. So this is another example in oat aphids, where we've treated aphids with these different fungicides, and some of them are killing the aphids, but others don't, but they do have effects on the next generation. So if you look at this figure in the middle here, that dot in gray is the untreated aphids, and then you've got the different colors with the aphids treated with different chemicals. And so some of these really are reducing the fecundity of these aphids in the next generation. And then this figure on the right here also shows that they reduce their survival in the next generation too. So just to sum up, endosymbionts have really dramatic effects on aphids, and they can potentially be used to our benefit. So you could potentially release aphids with different endosymbionts, and they could be used to reduce the populations of aphids or stop the transmission of viruses to crops by aphids. But unfortunately, we're really limited to the the biological variation that already exists. So all of these different effects of endosymbionts may not be useful, but maybe we'll get lucky and we'll find something that can be useful. And then, yeah, so we're also looking at different chemical treatments which can target endosymbionts. And, you know, we're thinking that these could be good alternatives to the current insecticides because they have longer term effects. And so I'll leave it there. I'd just like to thank everyone from the AgPIP team at the University of Melbourne. Yeah, thanks for listening.